Hello, and welcome to another episode of Max Planck Florida's Neurotransmissions Podcast. I'm Lesley Colgan, and I'm here today with my co-host, Dr. Akash Pal.
Hello.
How are you doing, Akash?
I'm doing great, and it's wonderful to have Mark here today so that we can talk a little bit more about introception and some other cool stuff he's been doing in his lab.
Yeah, thank you so much for joining us today.
I'm very happy to be here.
No, it is fantastic to have you here, Mark. You are a leader in the understanding of neural circuits and introceptions and how these circuits shape perception and behavior. So when it comes to introspection, how do you define it?
So when I focus on interoception, we think of it as the sensing of body signals. And think of this as sort of a central process that's happening all the time. So even if you're not going from one place to another, you're just sitting peacefully, you're still listening to your body, and that essentially informs almost everything that you're going to do in the future. Every time you take an action or you think about something, you're also caring about how your body is reacting, and that's informing how you process that information.
What kind of internal states are you currently focused, your lab is currently focused on that you're working?
Yeah, so we started the lab in a division of endocrinology and metabolism where people were really interested in hunger and satiety in the context of obesity. I came from basic circuit and cellular neuroscience in a more foreign land of the visual cortex. And so we met in the middle and focused on hunger and the neural circuits underlying hunger and how those neural circuits are also regulating what we're looking for in our world.
Mm-hmm. And then what is interesting is for your PhD, you worked with Chris Moore. Chris Moore was actually a committee member for when I was in grad school, and you focused completely on the barrel cortex, and now you have moved so far away from that. How has that journey been?
It's been actually a wonderful journey because I think, most fields really benefit from having a lot of crosstalk from people who have different backgrounds. And my background from moving from physics to then thinking about the barrel cortex and flexibility of cortical circuits and auditory and visual cortex, I really wanted to try to get at questions of flexibility and how what we need guides what we pay attention to and what we're looking for. And people were studying this in non-human primates. It was difficult to study this in mice, and then I started my job at the Beth Deaconess Medical Center, where we could collaborate with folks who would turn on neurons in the hypothalamus, make the animal hungry even after they just finished a meal, and they were looking around for the places where they could find food, and we realized that actually hunger and thirst and other fundamental drives and the associated circuits in the brain are a great way to understand flexibility, because what you need is changing all the time, and then that's going to recruit different forebrain circuits that are going to change how you pay attention to things in the visual world, in the tactile world. So ultimately, that is a way that I tell myself, at least, that I can bring together my past life and the sensory cortex and my current focus.
Mm-hmm. I was interested in, how you moved from your early training in biophysics into the neuroscience realm and into the idea that interoception and internal states sort of shape our behavior. Was there something particular about this topic that really grabbed your attention, or a particular experience?
It really kind of evolves gradually over time, and I would say not in a directed way, but really in a more opportunistic manner, making use of synergies with colleagues along the way. I realized when I was doing physics in college that the possibility of working in neuroscience was really the possibility of working with people who were much smarter than me at various levels, from basic chemistry all the way up to psychology, and that really drew me to neuroscience. And I think after when I was trying to figure out how I wanted to start my own lab, I really liked the idea of having a more naturalistic kind of embrace of basic motivational drives that are fundamental and linking that to sensation and perception. And it was really after studying the cortical circuits that are involved in these core drives, such as hunger, that we realized that actually, much as the rest of sensory neuroscience and hippocampal neuroscience was thinking about spatial contexts, that in the context of hunger or thirst, there was an associated body context. and that is all the sensory signals that are telling you that you're in a hungry state. That actually creates a context, and that the insular cortex, the part of the cortex that's receiving these bodily signals, is able to construct a representation of that context that can then structure and organize perhaps how the brain recalls associated, for example, visual representations that were useful in the past for getting food when I was hungry, or for getting, what should I look at to get water when I'm thirsty, right? So that's really how I kind of moved towards a focus on things like perceiving the signals from the body.
Earlier, I really wanted to ask you this question, especially after the seminar. That was a brilliant talk. So in the seminar earlier this morning, you outlined two different circuits that are both involved in hunger. One prefers satiety, one tells you how hungry you are. Do you mind telling us a little bit more about these two circuits and how they work with each other?
Sure. So there's been a lot of work from many labs, including Brad Lowell's lab, Scott Sternsen's lab, Jeff Friedman's lab, and others, that discovered neurons that are critical for not so much the feeling of hunger, but for how we react when we are in a starvation state. And we react by mobilizing all sorts of motor plans and all sorts of new behaviors that are going to allow us to restore calories. And an important one is the seeking of food and the suppression of competing drives. And that is achieved in large part by these agouti-related peptide neurons in the bottom of the hypothalamus. And these neurons are amazing. In mice, for example, that have just eaten a Thanksgiving dinner, if you turn on these neurons with chemo or optogenetics, the mouse will get up and eat another 10% of its body weight. And this was striking to me because it was very specific. And when you look at where these neurons in the hypothalamus are projecting, they're really regulating all sorts of circuits throughout the brain to achieve this goal in a concerted way. So these are the AGRP neurons. They're the neurons that are active when you're hungry. And they're sort of the yin to the yang of the neighboring neurons called these POMC neurons that are quiet when you're hungry but active when you're sated. And it's really sort of this intuition about a gas pedal and a brake that are working together that we think is really important as a sort of a general, I think, a common thing that you see in neuroscience where in order to change from one state, for example, from hunger to satiety, it's not always adaptive to just turn down the activity of one neuron type. It may be useful to simultaneously turn up the activity of another neuron type. And as I discussed in the talk, it turns out that that change in releasing the gas pedal and stepping on the brake at the same time turns out to be very important.
And then, so would you say there is some kind of a middleman between the gas pedal and the brake that communicates this?
Yeah, so there are direct connections between these neurons that drive activation of one to inhibit the other, but there are also many kind of almost beautiful ways in which receptors, such as the leptin receptor, will drive opposing changes in activity in these neuron types, and also in which other types of inputs, synaptic inputs and other hormonal and neuromodulatory inputs are differentially regulating these two neuron types so that they always seem to be moving in opposite directions. And we were sort of further drawn to these neurons in the context of thinking about hunger and satiety because these AGRP neurons seem to drop their activity when the mouse eats a bite of food in a manner that seems proportional to the caloric content of the food. So the more calories, the more these neurons change their activity. And that was really exciting, but it left an open question, which is, okay, so that happens with each bite of food, but how do you then go from that to coming up with an estimation of how many calories total you've eaten over the course of a meal? And we think that the downstream neurons that are listening to these AGRP and POMC neurons are somehow integrating all of this information in each bite in order to create a running estimate of how many calories have been eaten in the meal and whether or not I should continue to eat. That's still very speculative, but that's sort of where this work is taking us.
So in the context of, let's say, binge eating or obesity, this relationship between the gas pedal and the brake you mentioned, am I to understand that that is what is dysfunction?
That's a great question. So I think one of I remember when I was a kid and brakes weren't as good as they are now, and my dad used to go down steep hills by pumping the brake and releasing it and pumping it and releasing it so the brake didn't get too hot. Mm-hmm. And so one perhaps analogy of that is that, if you're in an obese state and you're sort of eating too fast, that you may essentially become less effective at bringing about a change in brain state that shifts you from hunger to satiety. And the work that I described today discusses the way in which the biological processes that are set in motion by taking a bite of food, even though that bite only lasts a few seconds, it's driving the activation of these POMC neurons, and this burst of activity in these neurons releases peptide that is sensed by a downstream neuron. That downstream neuron then creates this second messenger cascade. Essentially, it's triggering biochemical signals within that neuron, and those are sticking around for many, many seconds to a minute. And they're driving plasticity processes that we think are sticking around for tens of minutes. So the bite that you took for a few seconds has many seconds to minutes of impact on the brain. Now, what happens if you binge eat, right? You're not giving the brain enough time to process the first bite before another bite comes in. So we think that much of what we've heard from dietitians about the importance of slow eating may actually have its roots mechanistically in the need for the brain to kind of carry out its plasticity at a reasonably slow time scale.
If I'm able to manipulate these second messengers, either upregulate or downregulate, do you think that's a way to control binge eating? That would be a way to control binge eating?
That's a very, very interesting question, right? So if you could manipulate it, certainly when we've manipulated it directly in our mouse experiments, we can increase or decrease the rate at which the mouse becomes sated. And we think that some of the existing anti-obesity drugs are already tapping into that very same mechanism because they're acting on receptors that are regulating these signals. However, whether or not you could prevent binge eating in that way or decrease the consequences by that's an open question, I'd say. But I sort of see some of the medications that could regulate the biochemical signaling as something equivalent perhaps to a nicotine patch, right? The idea that maybe what you still need to do is work on behavioral strategies for improving your relationship with food and consumption, but maybe you can sort of make that easier by augmenting that with a pharmaceutical strategy.
More of a palliative fix than the core issue.
Yeah, I think that's right. Ultimately, changing lifestyle is going to be an important part of any ability to sustain a healthier relationship with food. I think what we don't understand is, for example, why the current best generation of anti-obesity drugs, when you remove them, why there isn't any persistent lasting effects in the sense of maintained lower body weight. It seems that there's a lot of rebound that happens right away. So if people were generating and able to generate a better kind of set of habits while they were on these drugs, then you would imagine that might have persisted more than it has.
Your investigations into understanding the neural circuits that regulate feeding behaviors has tremendous translational potential. And I actually was hoping you might touch on another one of your recent papers, which came out in Neuron about repeated stress in female mice and how it led to sort of a preference for them to seek what we could describe as a starvation-like state?
Yeah, I'd love to. This was really a labor of love, this project that was led by Hakan Kutsukdareli, who's a fantastic postdoc. The premise was as follows. We knew from excellent work from the Al-Hadef and Bentley labs that one of the many things that AGRP neurons, these hunger-promoting, food-seeking neurons, do in order to get you the calories you need when you're starving is to say, don't be afraid of venturing out to that watering hole in the middle of the desert, even though you have a strong risk of predation. Because if you don't get the food, you're basically toast anyways. So one of the things that AGRP neurons do is they suppress all sorts of things like anxiety and inflammatory pain and itch and other things that are going to get in the way of foraging and seeking the food. And so we knew that from many studies from different labs, in fact, when you're in a food-restricted state, you are less susceptible to acute stressors. However, at the same time, there's been studies from the Sternson lab and others suggesting that actually the state of having elevated activity of your AGRP neurons, the state of being food-restricted, even though in healthy animals it has this ability to suppress pain, itch, and so on, it's still aversive. If you give a mouse a choice when it's healthy of turning on its AGRP neurons or leaving them off, healthy mice will choose to leave their AGRP neurons off. And this is thought of in the context of a word called drive reduction, that you have these aversive drives and you want to kind of get rid of them. In Portuguese, Brazilian Portuguese, like it's basically like killing your hunger is a common idea. Okay, but suddenly now you're in a context where during early life stress or genetic predisposition to anxiety, you have this very high level of anxiety and sensitivity, for example, to pain. In this situation, we hypothesized that actually AGRP neuron activation may actually be the lesser of evils and that you'd want to actually turn these neurons on and thereby suppress the anxiety, which is otherwise out of control, even at the cost of the feelings associated with starvation. And so, this is essentially the concept of sort of a self-medication, a blunt hammer that allows you to gain the positive benefits of being in a starvation-like state with the side effects of just feeling starving and basically also having metabolic dysregulation. So we set up a quick experiment at the beginning, which wasn't quick 5 years later, where we instead of actually trying to see whether the mouse would food restrict itself, which was a difficult thing to do and we wanted to avoid, we just asked the mouse whether or not it would prefer or avoid rooms in a virtual reality environment that were associated with turning on its AGRP neurons. So this is akin to what people think of as a real-time place preference, but a choice to actually turn on certain neurons in their brain or not. And as predicted, especially in males, but sometimes in females, in healthy animals, you saw the animals avoiding AGRP stimulation. But after we induced a chronic stress paradigm for many days, what we found was quite striking, which is that a subset of the female mice, and only the female mice, suddenly developed an incredibly strong preference for AGRP neuron stimulation. They really wanted to put themselves in chambers that made them feel like they were in a state of starvation or a starvation-like state. And in fact, we could predict which of these mice decided were going to become those AGRP-seeking mice a month before the whole task, just by their degree of, let's say, sensitivity to anxiety, even kind of in a separate test. So, what's interesting about this is that it's I think a kind of a flavor of preclinical mouse model of a psychiatric disorder that can capture aspects like the sex specificity and the trait predisposition by focusing on specific aspects of the psychiatric disorder. So late stages of anorexia nervosa are obviously impossible to model, I think, in mice, body image changes and so on. This is really saying that actually maybe many of those psychiatric disorders are rooted in more fundamental issues of how do I find behavioral strategies to decrease my anxiety, and what can trigger habits at the beginning of that seeking of a less anxious state that then become habitual. I think that's the kind of thing that's studyable in mouse models for many other anxiety-related disorders, even if we can't really go all the way to the other end.
So what you're saying is, let's say I have an experiment in the morning and I'm very anxious, I should skip breakfast.
So, I mean, I think that's an interesting point. What I'm saying is don't do that because it might become habitual and maladaptive. I think that what I'm saying is try to have breakfast with your friends and your family and find other sources of stress reduction that prevent you from making these kinds of choices.
So throughout your career, I feel like you've really been sort of pushing the boundaries in terms of technology and adapting new technologies into what we can actually see and record from the brain as it's actually working. I wonder if you could just take a minute to sort of reflect back on, how technology has changed since the beginning of your career, and then maybe give us a look to the future.
Mm-hmm. Such a great question. I was trying to decide what I was going to do my postdoc on in 2006, 2007, and I had been familiar with electrophysiology before it expanded to these 1,000 contact probes. And I remember I was in Clay Reed's lab one day, and I was looking at this microscope, and the postdoc was turning a dial, and you could sort of move up and down and see these different neurons light up. And I swear I remember so clearly the sense of awe, but also the sense that even though all of the sensors that were available at the time were rather crude relative to where they are now, that this was the beginning of something where if one could do what they could to get things moving at that point, that all of these other amazing labs that were developing kind of sensors and new physics for improving the microscopes, that this was actually going to be something that could just expand indefinitely. And that's exactly why I chose the imaging. I just loved also just the feeling of being in those experiments and participating, but also this sense of kind of endless expansion of what we could do. And here we are, almost 20 years later, with just a tremendous library of their sensors, not just for calcium as a proxy for spiking, or even sensors of membrane potential itself, but also sensors for all kinds of neuromodulators and peptides, and sensors for metabolites, sensors for ATP, and the list just goes on, to the point where really there's nothing other than somebody's imagination that would limit their ability to create a new research program in 2024. And so, looking to the future, I think one of the things that has been a lingering limit on trying to make progress in understanding biology is that even though we have all these sensors and actuators, I mean sort of things that can turn on or off neurons or turn on or off second messengers, that we only are limited to imaging or to stimulating in one or two colors, and that's really in contrast to nature and the brain, which it involves hundreds of molecules that are changing across motivational states that are being integrated by a neuron at the same time, and then also hundreds of changes in the release of those molecules. So unless we understand a more holistic integration of the chemicals that surround a neuron at the level of tens to hundreds, we're going to miss on this critical computation that an individual neuron is doing when it's integrating its surroundings. And so one of the directions that I'm excited about that our lab has tried to work on, Peter Kalugan in the lab's going to post a bioarchive on this soon, is can we create technology to record from 10 or 20 sensors at the same time. These would be optical sensors. And in this way, try to have real-time tracking and eventually closed-loop control of many different signals at the same time. And I think there are many parallel technologies that are moving in this direction, and I'd just really encourage sort of younger scientists in the audience to consider the idea of moving away from just tracking or manipulating one quantity at a time or one signal at a time. The brain definitely is doing a lot more in parallel, and we need to understand that.
It is a nice segue. If we could go back to your talk one more time before you started the talk, I don't know if you want to talk about it. You talked about a green lens probe with a hydrogel matrix at the end that has different sensors so that depending on the local environment, it can tell you what these neurotransmitters and ligands are. Correct?
Right.
All right. So as you were telling me about that at the talk, what I was thinking, do you think it's possible to make a neuropixel-like probe but with sensors? What I'm thinking is, let's say if you have a very thin cylindrical electrode-like with gratings on the side, and then you fill the whole thing with hydrogel. And at different levels, you have different hex cells with different sensors. And then this way you have a biosensor-like probe. Do you think is that something that you're working on?
We're not working on it, but I think there are many different strategies. So there are electrochemical methods using aptamers, the same aptamers that you might have heard of in the word SomaScan, or various ways that are then being transferred into electrical charge that can be detected. What we like about using these G-protein coupled changes in fluorescence as a reporter is that the G proteins are already optimized to kind of the natural biological range of these molecules, and it's really a real-time calibrated process. So we developed this gel-based platform that could house 10, 20 different sensors, dip their grid lens in the brain, record the local chemical environment, and then take the grid lens out of the brain and dip it robotically into a 384-well plate of standards in order to know which sensor on the front of the grid lens is expressing which optical sensor. And in that way, we can get around this problem of limited number of colors by having all of the sensors be green, but being able to functionally identify which one is which. And to move towards another important consideration, which is the importance of being quantitative about concentrations, because we know that the concentrations and the quantitative levels really are going to impact whether or not our receptors can actually see the concentration, whether it'll be saturated, whether it'll be too low. Your suggestion is, can we move beyond that to a probe where each individual region of the probe, for example, has a different let's say hex cell or cultured cell expressing, or even a liposome expressing different fluorescent sensors. And I think there are many ways that you could come up with that. I think that the idea of, for example, kind of these printing technologies for printing onto, glass in very small quantities, or printing onto, they have these sort of fiber bundles also that have thousands of fibers. But in principle, if you could print onto each of the fibers a different sort of sensor cell, then you could definitely just basically use a standard CCD camera and read out each pixel would just be a different sensor. And so to me, I think this really is a valuable direction to move in the future. I think it's a great idea, and you should come and try it.
Oh, definitely. I would love to. But, it definitely, with the way that science is progressing these days, it definitely takes a village, right? Not one person can do everything. Yeah. Can you tell us a little bit about your lab, how big it is?
So the lab is kind of an amazing place. I'm so grateful to get to work with these people. One of the lab, we have about 6 or 7 actually, at the moment, it's closer to 5 postdocs and, about 5 graduate students. And then we have several research assistants and full-time undergrad co-op students and a lab manager. And so there's a lot of, I suppose, challenges of having a lab of about 18 to 20 people. But one of the wonderful parts about it is that you can afford to have sufficiently diverse range of expertise that people really know that they can ask each other for help, and also establish new collaborations kind of on the spot without having to always reach out beyond the lab. So for example, on this project, it was actually a collaboration with Maria Lehtinen's lab, and they have a lot more experience in molecular biology and in culturing the cells. But we also had people helping with a lot of the optics. We had, at the end, these gels were, warping in horrible 3D ways, and so we had to be able to align and, basically if a brain, I don't think there's ever been a piece of brain that's warped as badly as these gels warped. So all of the algorithms that were used for aligning and nonlinear registration of brains really started to fall apart. And so we were using AI and all kinds of different machine learning algorithms. And it's kind of amazing that it all came together. And it's really a testament to both Peter Colgan, who's driving it, but also to this amazing team of people that he had helped assemble to help him with this. So I do think that where science is going is towards a more and more team science approach. And it's clear in 2024 that a team science approach is challenging because there's still the sense of individual recognition that who is listed first on a paper and how do we know what it means for someone to be listed third in terms of what they really did. And it's outside of the PI's control to some extent, other than really being an advocate for everybody and their contributions when it comes to writing a letter or picking up the phone and advocating for someone for the next step in their career. And it's much more complicated in terms of designation of authorship. It's much more complicated in the sense that sometimes people have perfect complementary expertise, but they also just don't perfectly match, right? And so all of those things require basically a level of just creating an ecosystem where things that evolve organically at the beginning of the collaboration, and enough also support so that people also feel like they can try to create new things because they're also getting support on other aspects of their project. So I think it's much more challenging than having one person, one project, but at least in my view, it's been very satisfying because a lot of the entropy, the chaos that's created by these team science projects ultimately lead to individuals within the team stepping up and developing leadership skills and corralling everything and everyone into helping them get what they need. And that's the best thing you could learn from an experience in the lab in terms of being prepared to being an independent investigator.
Dr. Andermann, I think we're out of time, but thank you so much for sharing your perspective with us today. I know I'm excited about the future.
I'm excited too. Thank you very much.
It's been a pleasure. Thanks very much.
Thank you.
As always, thank you for listening to Max Planck Florida's Neurotransmissions Podcast.
If you enjoyed this episode and would like to help support the podcast, please subscribe and tell your friends.